All right, what we have here is our benchmark on mitosis, all right, where we are going to elaborate more specifically on what's going on in this portion of cell division. So, mitosis, or the end phase of the cell cycle, is really referring to nuclear division. All right, this is when one nucleus becomes two nuclei. It has nothing to do with the actual cell itself dividing yet. That was that cytokinesis that we already talked about. Instead, this is talking specifically about the nucleus, all right, dividing. We're really talking about PMAC, all right? Interphase and cytokinesis are their own steps, separate. So, our four stages are prophase, metaphase, anaphase, and telophase. Prophase, it's going to be the first step. What's going to happen is that your chromatin, or your DNA, all right, so by chromatin, we're talking about those strands of DNA. Remember, they were copied, they were replicated in the synthesis phase of interphase, right? So all of those DNA strands are going to coil up, and now they're actually going to be visible chromosomes. Whereas before, they were just squiggles. Now they're going to be all very, very tightly coiled up and thick so that you can actually see them. But keep in mind that they were already joined at that centromere, right? They were already joined because that happened during interphase. In addition, you can see here that the nuclear membrane is starting to break down or starting to dissolve. There's no more distinct dark area where the nuclear membrane should be. Instead, it really just kind of looks more like a fading membrane. As well as the structures called centrioles. Right? If you guys remember, the centrioles were those structures, and a lot of you guys told me that they look like churros, right? Or I said they look like little Twizzlers. Some of you guys said they look like penne pasta, and they sat perpendicular to one another, and there were two of them. Right? They're actually going to start to move to opposite sides. There's a copy, all right? Now there are four of them, and they're going to start to migrate to opposite sides. And as they move to opposite sides, these things called spindles are forming. They're small fibers, all right, that are going to help to move the chromosomes during this division process. So if you look, here are two centrioles, right, in this picture, and you can see that they're kind of starting to move to opposite sides of our cell. Animal cells, the centrioles are moving to opposite sides because we have centrioles in animal cells. However, in plant cells, there are no centrioles, right? So there's not going to be any centrioles forming any spindles. Instead, all right, there's going to be a different fiber that's going to perform the same job. Don't worry about the specifics, but just remember to keep in mind that plant cells do not have centrioles for cellular division. Metaphase, all right, our second step, all right. I typically keep meta in mind because meta, middle, all right. Meta is middle. And in metaphase, the chromosomes line up in the middle of the cell. Here are our centrioles. These fibers that you kind of see, those are those spindles. All right, and the spindles are going to attach to the centromere, that middle portion of each of our chromosomes. All right, they're going to attach into that middle portion, and they're going to play tug of war, pulling on the chromosome until it lines up right down the middle with all of its other chromosome friends. Right? They're easiest to identify at this point. It really looks like a big, giant, massive cells in the middle. All right? You're not going to be able to make out each little distinct chromosome in that X-type shape. All right? But you're going to distinctly be able to tell. I mean, look at the thickness here we have of these chromosomes. So this is a graphic picture. All right, kind of showing you more along the lines of what you're understanding in your mind. Anaphase. In anaphase, I refer to ana, apart. All right? Ana is going to be the third step of PMAC, of mitosis. In this case, what's actually going to happen is that, remember how I drew you that picture of those spindles attached to the centromere of the chromosome? What they're going to start to do is they're each going to start to tug separately in opposite directions. Or you can think of it as the centrioles are like reeling them in, like a fishing reel, all right? And they're pulling on this string, and this string is tugging and tugging and tugging it. The chromosome is going to tear in half. All right? Inside, we have one strand of DNA on this side. Its identical copy on that side. One chromosome, sister chromatids. Remember, chromatids have to be identical copies. Right? That's how we use that word chromatid. Identical copies, and they're joined. So what's going to happen is those sister chromatids, all right, are going to be separated at the centromere. And as they're separated, they're going to get dragged to the opposite sides by the spindles. A lot of times, to me, all right, when we look at it under a microscope, to me it kind of looks like a bunch of fingers, all right, in each side of the cell. And those fingers seem to be like being pulled away from each other. All right, now we can go back to calling each of those separate strands a chromosome, because chromatid, we only use it when we're referring to the attached version. Here's another diagram. Telophase, our last stage. If it helps, I think about like a telephone, how a telephone kind of typically has like, you know, one end where you speak into, one end where you listen to, and I don't know if that helps, whatever. Oh, that's a typo. This is the fourth phase of mitosis, not third, sorry. Fourth phase of mitosis. This is the last phase of mitosis, of nuclear division. All right, in this case, the chromosomes are on their opposite sides. The spindles have detached, they're starting to dissolve. You can see how we seem like we're kind of starting to go back to normal. All right? Instead of us being in this coiled up stage, we're going to start to go back to the stringy form. All right? We're going to start to go back to the invisible form of DNA, where it's not really visible by a microscope. If you look here, you can't make out distinct chromosomes anymore. All right? It just looks like there's a whole lot of stuff right there. And in this case, a new nuclear membrane is going to form around each separate set. But each separate set, remember, should be identical. Now that we have two separate nuclei, the cell will start to pinch in. All right? It'll start to dimple in, until it dimples completely, and we have two separate cells. Right? And in that case, cell division is complete after cytokinesis. So, our whole overview of cellular division, we've got interphase, mitosis, cytokinesis. We can take interphase and break it down into its three separate steps. First growth phase, synthesis phase, second growth phase. Right? Mitosis can be broken down into PMAT, prophase, metaphase, anaphase, telophase. And our last thing is cytokinesis, where the cytoplasm divides. We need to keep in mind the cytokinesis, we've got a cleavage furrow for animal cells versus a cell plate for plant cells. So we have I, P, Mat, C. I pet my angry tiger carefully. That should wrap it up pretty much for cellular division.